In this video, we will discuss about tunability of the CAP system with host immune response. So before we going into the today's topic, let us quickly revise the inducible CAP system that they have developed, as I might have explained it wrongly in our previous video. So in this case, what will happen is that when the KFC genes are uh, transcribed and translated, then it will form heparosan. It will lead to the formation of CAP. So how they designed the system is that this KFC gene formation is controlled by this lag promoter. So when this lag promoter are formed, then it will prevent this KFC genes formations, and thereby there will be no CAP formations. And when the IPTG is introduced, then it will prevent this lag formation translation. Thereby these KFC genes can be translated. So before IPTG, let us understand without IPTG conditions. In that case, the lag promoter is formed. It will prevent these KFC genes to transcribe and translate, and the heparosan will not be formed, and there will be no CAP formations against the around the bacteria. When the IPTG is introduced, then it will inhibit the lag1 promoter translations, but and in that case the KFC genes can be transcribed and translated, and the CAP formations will be formed around the bacteria. So this is what the CAP system, how the CAP system works. So how they check the tunability in that case is that when they use their iCAP system in the human blood along with this blood bacteria, and then check its survival rate at different IPTG condition, at different IPTG concentrations, and they are able to see that when there is no IPTG, the level of the amount of iCAP is below the level of detections. But when they use the IPTG, they can see the increase in the amount of their bacteria. And these things they have also shown in the panel B, where we can see that even with the dilute concentrations of the bacteria injections with increase in the IPTG concentrations, they are able to see more bacteria formations compared to the case where there is no IPTG. Now they wanted to check the clearance time. So when there is no KFC gene in the ECN, they are able to see its clearance time is very low, and it goes below the level of detections in less than 1 hour. But when they use the IPTG, they are able to control the amount of time the bacteria remains within the host. And in the wild type case, even after 6 hours, they are able to see stable ECN concentrations, indicating that their tunable CAP system can be controlled and can be cleared off when it is not been induced by IPTG. Now they wanted to check its response against the host immune system. So in order to do that, they co-cultured their macrophages with the CAP system, with induced and without induced with IPTG. So when it is not induced with IPTG, they are able to see more cell lysis. But when they are induced with IPTG, they are able to see less cell lysate, indicating that the CAP around the bacteria is able to prevent the immune response. And these things they are quantified in the panel F, where we can see the more apoptotic signals without IPTG and less apoptotic signals with IPTG. Now they wanted to check the bacterial immunogenicity through the cytokines that are released when the macrophages are co-cultured with iCAP system, and they check the 3 different cytokines. First one is TNF alpha, and they can see the TNF alpha cytokines are significantly higher in the case where ECN is used without KIFC gene, indicating that since the CAP is not there to protect the bacteria, the immune system can detect this kind of bacteria without CAP and generate an immune response. But in the case where ECN is capped or CAP is induced using IPTG, they are able to see less cytokines, and this could indicates that the CAP is able to prevent the immune response from the host against the bacteria. And these things, similar results they have seen it for the other cytokines that are presented here, that is interleukin 1 beta and interleukin 6. This will altogether indicate that their tunable IQ system is not only able to control its half-life time within the host or the circulatory time period within the host, but it can also control the immune response, so that it can have enough amount of time to perform its drug delivery at the site of tumor. So far we have discussed the tunability of the CAP system and the host immune response in this figure, or results that are presented in this figure. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.